The other sources of drugs on top of plants are also animals. Okay, animals have been studied for years as a potential source of drugs. Imagine in earlier days before 1920s, having diabetes was taken as a death sentence because there was no medicine to treat diabetes before 1921, as a result of the amazing drug discovery of the scientists called Bantis and this guy, very interesting guy, Frederick Banting and Charles Best, who tried to isolate insulin from an animal. Imagine. Since then, now you can imagine now it has really revolutionized the management of especially type 1 diabetes and also type 2 diabetes with insulin. Not only that, the drug like heparin is also isolated or discovered from animals, and that's why heparin has really helped in managing quite a number of coagulation disorders, coagulation during pregnancy associated with different medical and surgical procedures. Is an amazing anticoagulant medicine. So yes, we have appreciated plants as a potential source of drugs, but animals are also an amazing source of drugs for years. Nowadays, the drug discovery process has a bit shifted, and I'll talk about it in the next video, where even the discovery of insulin, the discovery of heparin, nowadays we are not really sacrificing animals to get insulin or heparin. Now, biotechnology, engineering has really picked it up, and now we have quite a number of more modified and better medicines, because this is animal derived medical products. Usually are characterized with an allergic reactions when they are given to humans, because they will have an animal protein and the body will have an immunological reaction to it. So nowadays, most of the antibodies, most of these products that used to be isolated and discovered from animals are now becoming humanized, meaning that they have little or no reaction with regard as compared to those animal products. But drug discovery from animals has really shifted the thinking of drug discovery, and also has really solved quite a number of illnesses. In semi-synthetic, as the name implies, scientists will take the naturally obtained medicine. Take, for example, penicillin. Initially, Alexander Fleming isolated penicillin from mold, but you see the first penicillin had a number of problems. It was not soluble, you can't give it orally, and also it was only working against gram-positive organisms, okay? That is why now further they took and then it was modified. Now the chemical structure, the chemical characteristics of penicillin was modified, and now we have drugs like ampicillin, amoxicillin. These ones are what we call semi-synthetic, which is medicines which have been a bit modified from their initial or naturally derived way. That is another option. Also, we have synthetic medicines where we just go to the lab and you just try to synthesize, and those who know chemistry will understand this very well. You just synthesize. Take, for example, paracetamol. It's just synthesized in the lab. You can take aspirin. You can go to the lab and you can mix salicylic acid and also other chemicals, and then you can get aspirin, which is acetyl salicylic acid. So we have a semi-synthetic and also synthetically derived medicine. So what are we saying? So the drug discovery source of medicines could be plants. Some drugs you'll find them in the market isolated from animals, or maybe through the years, scientists have really modified them, or maybe we have a semi-synthetic, or some products are purely synthetic, those designed in the lab through a chemical reaction. What an interesting thing to be in the space of drug discovery, solving problems, solving illnesses using powerful drugs, which later will be now medicines. How are these drugs really discovered? We've seen in the earlier 3 videos when we talk on source of drugs. We talked all plants, animals. We say drugs could also be isolated or discovered in the laboratory using semi-synthetic or synthetic process. But how? What is the process? We have quite a number of processes which we can follow to discover drugs. The first one is called an in vitro approach, which I'll explain shortly, which is drug discovery using in a dish or in the lab. The other one is in vivo, drug discovery in a living organism, maybe in animals. We also do discover drugs in silico. In silico means silicon or in a computer. I'll talk shortly. And also we discover drugs ex vivo. We try to check whether the drug works or not in isolated tissue. And when all these evidences from the different pieces match, and when we have the proof of concept, again, we take it to the clinical trial. In the next series of videos, I'll talk about each of them in a bit detail. See you.